So in the previous video we were mentioning the fact that you have a lot of DNA molecules inside your cells, a lot of nucleotides, because we have so many nucleotides and genetic material. And we also mentioned the fun fact that if you take all of the genes inside the DNA inside your cells all over your body, you will be able to reach all the way to the end of the solar system and back. But how do we actually form and hold and contain this amount of genetic material inside each cell? We do it through a way called DNA compaction. And DNA compaction is a way where we take the DNA double helix and we dense it, we call it into a more organized form. And in this lesson, we are going to go one by one through the level of organization, and we are going to understand which are the level of organizations that are very high yield to remember for the admission exams. There are many different levels of organization through the way that we are not going to mention, but please know that we are only going to mention the very important high yield parts of the level of organization. So we are going to start from this illustration. This illustration shows first of all the uncoiled DNA double helix. Then we see the coiled double helix that goes all the way to something called histones. Now histones are proteins that can be formed and can interact with the DNA double helix in order to form a more dense form of the DNA, which is the first level of organization, through a structure called a nucleosome. So a nucleosome is where we take 8 histone proteins and we wrap the DNA twice around the 8 histone proteins. And I would like to zoom in on one of these following nucleosome. And as you can see from this picture, we have something that is pretty complicated and with many names, and I'm going to explain everything step by step. First of all, I would like to say that for the admission exam, you shouldn't memorize the names. It's very low yield for the BMAT and the IMAT exam. It's very low yield to memorize the names of the histones proteins. What you do need to remember that we have 4 pairs of histones and we have 8 in total. And when we take the DNA double helix strand and we wrap it around twice, we reach a point where you have a nucleosome. And this is the definition of a nucleosome. In the previous lesson, we were analyzing the nucleotide itself, and the DNA double helix itself. I would like to do it again, and I would like to add another very important property of the DNA that is going to be crucial when it comes to the actual condense or in the compaction of the DNA. So if we take and we go to the slides of the previous lesson, we are going to see this backbone that we have over here of one strand of the DNA. The other one, of course, has the same backbone. If we zoom in, we are going to see that the phosphate groups are negatively charged. And because we have so many phosphate groups inside our backbone of the DNA, overall the DNA is negatively charged. What does it mean and what is the significance of the charge of the actual DNA molecule? It's very, very important when it comes to the condensation of DNA. So histones are usually positively charged protein. And when we take a negatively charged particle and we want to interact it with a positively charged particle, what we are going to see is that the particles are going to attract each other. Therefore, they are going to be likely to compact together. So how does the nucleosome form? It is formed from histones that are positively charged and the DNA that is negatively charged, and they attract each other, therefore they form the following denser form. The next form of the DNA compaction is where we take the nucleosomes and we put them together in a structure, a more dense structure, which is called a chromatin. A chromatin is multiple nucleosomes that come together. Now we have two types of chromatins that are very important and high yield to remember for the admission exams. One is euchromatin. Euchromatin is the less dense chromatin form from the two that we are going to discuss. And when you have a less dense form of chromatin, we can actually transcribe this part of the DNA. However, heterochromatin, on the other hand, is a very dense form of DNA. And when we have a very, very dense form of DNA, the molecules that actually transcribe and make the RNA and therefore the protein from this area won't be able to access this area because it's so dense and together. So heterochromatin is not transcribed and euchromatin is transcribed. And it's very important and high yield to remember. Now, the next level of organization is just where you have the further condensation of chromatin fibers into a further level of organization. The next one, which is the most high yield thing you need to remember, is that the most compact DNA structure level of organization is called the chromosome. And we have chromosomes only when the DNA is compacted to the most compact level. Then it is called and defined, the definition is chromosome. Now these are the level of organizations that are the highest yield to remember. In the next lesson we are going to actually understand what is the process of epigenetics and why different parts of the DNA are actually transcribable and why other parts aren't. Because we talked about the euchromatin and the heterochromatin, but we didn't talk about the chemical reaction that actually occurs in order to create different parts of the DNA in order to give proteins and RNA and proteins. Because in the previous videos I was mentioning the fact that you have many different types of cells inside your body. However, each cell contains the same exact book of DNA, the same exact instructions. The only thing that changes is which pages from this book, which proteins and which RNA are going to be transcribed and translated from this DNA. And we do it through a process called epigenetics, is where we allow the transcription or disallow the transcription of different parts of the DNA through the process of epigenetics. So in the next video we are going to touch epigenetics and the chemical modifications you can do to the following molecules in order to allow or disallow the transcription of different parts of the DNA, therefore give us different types of cell inside our body. I will see you next time.